Good afternoon, everybody. This has been exciting days for Immunicum, as this morning we announced the results of our phase 2 study that I will show you briefly. So unfortunately, I've not been have much time of spending with you here at the conference. So we are a publicly listed company, NASDAQ Stockholm, in the main market. We operate in the space of immuno-oncology, and we do that with a cell-based therapy with some key differentiations. It's an off-the-shelf product, is originated from allogeneic cells, and we do activate the patient immune system. We are already quite advanced. As I mentioned to you, we just announced today the results of a phase 2 study controlled in patients with kidney cancer. We have an excellent safety profile in over 90 patients treated in different types of solid tumors. And we have a very good manufacturing process in Germany, and now in the process of transferring it and starting activities for a commercial process in the US. We have IND open since 2016, already 2 studies running in the US, and of course long interactions also with European authorities. We enter into a collaboration with Pfizer, Merck, Germany, where they will supply us for free the checkpoint inhibitor for a new study that we have initiated at the beginning of this year in combination with checkpoint inhibitors. We raised last December 351 million SEK. More than half of it was through a directed issue, bringing big institutional investors like the AP4, AP2, Gladiator, Adrigo. So big institutions that have been very important in providing us, a longer-term perspective. That cash, together with the cash that we had in the company, provides us a runway towards the end of 2021, what of course carries us across big value inflection points, namely today and also next year. So briefly, what is ilixadencel, our lead product? Cells originated from healthy donors. These are blood donors, nothing special. They just need to be healthy individuals. We don't do any genetic manipulation, any expansion. We just activate these donor cells to make them extremely pro-inflammatory. When they start producing chemokines and cytokines, we cryopreserve these cells. So from one healthy donor, we treat up to 50 patients. We already passed 3-year shelf life with our product, so we really can manufacture, store, and the product is ready to be available whenever needed. Another key advantage is that we don't need to do any matching. The product is the same regardless of the patient or the type of tumor. So when it's needed, when the patient needed, just go to the hospital pharmacy, bring a vial, thaw these cells, and then you inject into the tumor. We can inject the primary tumor, we can inject the metastasis, it doesn't matter. You just need one mass. And when these donors, the dendritic cells that were activated, are injected into the tumor mass, they start producing these chemokines, cytokines. Massive recruitment of patient-owned immune cells, because that's one of the key challenges for the majority of the solid tumors is that these tumors are very cold tumors. They don't have enough patient tumor cells present. NK cells recruited to the site interact with the tumor cells, kill tumor cells, release neoantigens. That is the barcode for that tumor. Patient dendritic cells are also recruited to the site of the injection, take on these neoantigens, are matured faster by the production of interferon gamma from these donor cells, migrate to the lymph nodes, and they educate the CD8 plus T cells. And these are the patient-owned T cells that then are tumor-specific and enter into circulation and destroy the tumor cells. So basically, we operate on the left side of this tumor immunity cycle, activating the patient immune system, perfect to be used in synergy and in combination with the classes of drugs that are more on the anti-immunosuppressant side, the checkpoint inhibitors, TKIs, even some chemotherapy. So we are not going to be in competition with these big classes. We are going to be used in combination. Kidney cancer data today, I will show you that. We also have data published in liver cancer and in GIST, orphan disease. The new study that we have initiated is in head and neck cancer, non-small cell lung cancer, and gastric adenocarcinoma in combination with checkpoint inhibitors. The study is currently running in the US. We have now 4 studies concluded. Mareka, the phase 2 results announced today, and I will describe this to you, but it's 88 patients, 58 in ilixancel in combination with sunitinib, and 30 patients in the sunitinib-only arm as a control. So very, very important study. Kidney cancer, we published the results in January. Very strong data showing the immune activation, continue excellent safety. We are looking now for ways of continuing developing that indication. Very recently, we also announced the results in GIST. This is an orphan disease, as I mentioned to you, and these patients don't have many alternatives. They are basically changing from kinase inhibitor to kinase inhibitor, and very surprising and very positive. In 2 out of these 6 patients, 2 patients that were already progressing on the 3rd line of TKI responded and had a regression in the tumor. So very exciting data. Of course, a small number of patients, but very important. We already have studies. The first study in kidney cancer, we have a small number of patients, but we reached the 4-year median overall survival with patients still alive after 5 years. This is quite an extended survival, when the expected survival with sunitinib is 14 to 16 months. So let me give you a little bit of the key top-line results we presented today. So one of the exciting parts of the data is that we achieved complete tumor responses. So in 5 out of 45 patients, 11%, these patients got totally rid of the tumor. And this is a totally unexpected result, as a positive. Investigators and clinicians said that it's very rare that in these very sick patients, because these were all metastatic at diagnosis, you will have these type of responses. We still have, at 18 months, 2/3 of the patients still alive. So from a perspective of median overall survival, very positive. The patients continue alive. We need to continue following them, and we will do this at 6 months interval. And most importantly is that, compared with other of the recent approved combinations, we really didn't bring any additional toxicity, safety concerns for the combination. This is an exception because all of the combinations recently approved, both drugs when combined bring additional toxicity. So very important. As you can see there, from one of the investigators, this is rare, really rare that patients in this advanced stage have these type of responses. So of course, as I mentioned, a lot of patients are still alive, and we will continue following them. But this data is very strong and clearly supporting the continued development in this indication. And you can see it here just as a reference. The sunitinib group had 1 out of 25 patients, so about 4%. And this compares, if you see other studies, you have complete tumor responses in the sunitinib arm in about 2%. But when you look at the complete tumor responses in the combination arms, nivo and EP, Keytruda and axitinib, it's about 7 to 9%. Of course, there are bigger number of patients, but just to say that, getting these type of responses is quite encouraging, quite exciting, and definitely supporting the continued developing of the product. But we are also initiated a study, as I mentioned to you, so we're really diversifying the type of tumors that we are treating and the type of combinations to really maximize the value of the product. So phase 1b, head and neck, non-small cell lung cancer, gastric adenocarcinoma in combination with pembrolizumab, Keytruda, in the US. First patient started to be enrolled in February. This is a staggered phase in the beginning. And when we move to the phase 2, then these 3 indications will run as 3 separate studies. In head and neck and gastric, we will be combining with avelumab, that is the Pfizer checkpoint inhibitor, and in lung, we will be combining with Keytruda versus the CPI alone. So also very important data, and where, from a mechanism perspective, we have a very strong synergy that we have also already shown in preclinical studies. And again, we are going to start getting the initial safety data, and if we maintain our excellent safety profile, this is a very important piece of data because, again, most of these combinations have added toxicity. So when you look at the future and the potential, so we see that we have combinations with kinase inhibitors, sunitinib, sorafenib, regorafenib. Sorafenib, very importantly, we found out that it's not a good TKI to combine. A very important piece of data. Now we have the data in the new study with the CPI. And of course, moving forward in renal cell carcinoma patients, really different alternatives. The new standard of care is becoming the CPI plus axitinib. And of course, here we have the possibility of, in first line, to add to this combination because a lot of patients don't respond or have very high toxicity. And, with our excellent safety profile, this is one of the avenues. Of course, we can go a second line to the patients that don't respond to this combination. And we also have, additional types of tumors where we have the potential to do additional combinations. So really a lot of potential. The data that we have been producing in combination with different classes of drugs in different types of tumors really provide very good data to then move forward with the development of the product. The next step, of course, will be, and we will start working on that for running a pivotal study based on this strong data that we received. So to finish, so we have quite a unique positioning. We are the only ones with our approach. It's quite innovative and immune primer. We have the big key differentiation that our product is an off-the-shelf product, totally different from other cell-based therapies. We have already strong data showing the synergy with different classes of drugs. We have now completed the phase 2. We have excellent safety. The side effect profile is primarily fever of low grade because we induce inflammatory response, and this is a key differentiation. We have initiated the activities to transfer the commercial process now to the US and to really develop a commercialization process. We have a very experienced team, relatively, with the company for the last 3 years, but our chief scientific officer is also one of the founders and inventors, so very important to have you join us, and staying with the company, of course. And very importantly, we have quite a nice runway towards the end of 2021, and the fact that we attracted a very strong group of institutional investors is also very important and give us, the runway to allow us to now. One of the activities we are going to initiate is the sharing the data with potential partners, as there is a lot of interest and not a lot of compounds that have a priming type of activity and the strong data that we have now shown. So thank you, and I'm happy to have any questions.